The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thanks for joining us today. On behalf of the New York State Federation of Lake Associations and the Syracuse University Environmental Finance Center, welcome to the webinar today on an introduction to lake ecosystems, a New York State perspective. This webinar is part of a series aimed to assist New York's lake associations and local government leaders with their skills for better lake management. Our experts today will be providing an overview of lake science and water quality management from the perspective of New York lakes. The New York State Federation of Lake Associations, or NYSFOLA, is committed to reaching our membership with important information. As it is for all organizations during this time of physical distancing, NYSFOLA's state and regional conferences have been canceled. While the opportunities to learn and network cannot be fully replaced, this webinar series does provide the opportunity to stay connected and continue to provide important information to our members. If you have any questions about NYSFOLA, please contact Nancy Mueller using the contact information shown, or you can visit the website at nysfola.org. NYSFOLA is pleased to provide these webinars with our co-sponsor, the Syracuse University Environmental Finance Center. And for a little bit about the SU Environmental Finance Center, our team enhances the managerial and financial capacity of regional and local governments, non-for-profit organizations, and local residents to improve environmental infrastructure, services, and quality of life. Our partnership with NYSFOLA for this webinar series is supported under a grant by a Rural Utilities Service, housed in the United States Department of Agriculture. We would also like to welcome all of the attendees today on this call to join NYSFOLA and the Environmental Finance Center for future webinars. Webinars will be held on Thursday mornings from 10 to 11 a.m. Registration is now open for one more webinar, so please visit one of the websites listed at the bottom of the slide for registration and more information. And during the webinar today, everyone will be kept on mute to ensure audio quality. If you have a question, please type it into the GoToWebinar question dialog box at any time throughout the session. We will be holding all questions for a Q&A at the end of the presentation. We will also be posting the presentation as well as this video recording on the Syracuse University Environmental Finance Center's website. You will be receiving a follow-up email that includes a link to all of these materials. Registered attendees can receive a certificate of attendance for participating in this webinar today. This webinar has not been submitted to licensing agencies for pre-approval of continuing education or training credits. If you would like to receive a certificate of attendance to apply for credit hours, please contact Tess Clark, pclark@syr.edu. And so on that note, I'd like to introduce our speakers for today's webinar. We have three presenters from the Upstate Freshwater Institute in Syracuse. I welcome Monica Matt, research scientist, David Andrews, research scientist, and David Matthews, director of the Upstate Freshwater Institute. So David, welcome, and I pass everything over to you.
Thank you, Savannah. Thank you for having us this morning and allowing us to talk to you today about limnology in general and about New York State lakes in particular. We have a lot to cover, so we'll dive right in. If I can advance the slide, there we go. I'll lead off, and then I'll be joined later by my talented, younger, and better-looking colleagues, Dave Andrews and Monica Matt. We'll talk about kind of a survey of New York State lakes, their origins, talk some about temperature stratification and dissolved oxygen depletion, discuss biological communities of lakes, also humans and how they can impact watersheds and lake water quality. Talk a bit about management and leave some time for questions and answers at the end. We've tried to sprinkle in some examples and case studies of including New York State lakes to hopefully make the presentation a little more interesting. Before we get into the main part of the program, I want to describe to you what the Upstate Freshwater Institute is, so you have some idea of who's talking to you. UFI was established in 1981 in Steve Effler's attic. We are a not-for-profit organization. Although we're independent, we do have close ties with academics from Syracuse University and from SUNY ESF. These academics constitute the majority of our board of directors. We have field staff, boats, field equipment, a water quality laboratory, and water quality modelers that conduct both applied and fundamental research on lakes, streams, rivers. Our mission at UFI is to provide the basis for the protection of the lakes, streams, and rivers of New York State, and also to, where we can, advance freshwater research and promote freshwater education. All right, we thought a good place to start might be kind of a bird's eye or satellite eye view of New York State, to get some sense for what the lakes are and where they are. I think remarkably, there are almost 8,000 lakes in New York. When we say lakes, we're including reservoirs and small lakes that some folks may consider to be ponds. And for most of us, we're familiar with the large, high-profile lakes, the Great Lakes, including Ontario and Erie, Lake Champlain, Lake George, the Finger Lakes, Oneida Lake, the thumb of the Finger Lakes. And maybe even those of us that are quite familiar with lakes in New York, maybe we could name 50. Of course, Nancy Miller could name about 7,000 of them, I believe. But for the rest of us, we can, we're really familiar with a lot of the larger lakes. And I think the lesson here is, although those are the lakes that we hear about and that may be well studied, the vast, vast majority of lakes in the state are much smaller and, for the most part, understudied. We are blessed with so many lakes, largely due to glaciation during the Pleistocene epoch that occurred about 2.5 million years ago up until about 12,000 years ago. And other states in the US that were exposed to glaciers, such as Wisconsin, Minnesota, Michigan, Maine, are also blessed with many lakes. These lakes were formed by the advancements and retreats of glaciers, which left deposits and moraines, which formed lakes about 10 to 20,000 years ago. The power of erosion dug out U-shaped channels in old stream beds that formed the Finger Lakes. Large chunks of ice that melted and left depressions formed many, many kettle lakes. There's some excellent examples here in the Tully area south of Syracuse. And in addition, humans have made many large reservoirs, including reservoirs in the Catskills that provide drinking water for New York City. And let's not forget about our fellow engineers, the beavers, who produce hundreds and hundreds of ponds and in large lakes across the state. So why is the origin of lakes, or otherwise known as geomorphology, why is that important? Well, it's important because it really establishes the physical, chemical, and biological character of lakes. It determines the size of lakes and their shape, how long water resides in the lake, or the inverse of the flushing rate, how rapidly water moves through. Chemical characteristics such as the ability to buffer acid inputs are determined by local geology, which also determines nutrient inputs for lakes or productivity. The biological community in lakes is really structured in large part by these physical and chemical properties. And as an example of the importance of geomorphology, we're going to compare Black Lake which is a lake in the northern part of the state, with Skinny Atlas Lake, which is located a little southwest of the city of Syracuse. Both very long, narrow lakes have almost identical surface areas. However, the contributing watershed area for Black Lake is approximately 10 times as large as the contributing area for Skinny Atlas Lake. So there's a much greater contribution of nutrients, et cetera, from the watershed in Black Lake. Also, Black Lake is only about 2.5 meters deep on average, compared to 44 meters for Skinny Atlas Lake. That's due to the lake bed being gouged out by glacier activity. As a result of these features, Black Lake is a very productive lake that supports a thriving warm water fishery, while Skinny Atlas Lake is cold, relatively unproductive, and supports a cold water fishery. In addition, Skinny Atlas Lake has amazing water clarity, about 7 meters on average, while Black Lake has Secchi depth less than 2 meters. There are different ways to classify the many different types of lakes in New York State. One is more of a regulatory classification scheme, which is based on best uses of a lake, which range from a drinking water source to perhaps a lake that is suitable for swimming. Other lakes may be only suitable for boating and fishing. Another way to classify lakes is by their productivity or trophic state. Lakes with low productivity tend to be deep and clear, and we call those oligotrophic lakes. Contrast with oligotrophic lakes are eutrophic lakes, which tend to be very productive, typically shallower, and may be prone to algal blooms. Mesotrophic lakes fall somewhere between these two extremes. And when we think about oligotrophic lakes and eutrophic lakes, we shouldn't think about that as good and bad. They're different. This is a natural progression that occurs over geologic time, and these different lakes can provide different types of uses for lake residents and people that use lakes for recreation. A central feature of lakes is that they stratify thermally. So during the summer, less dense warm water rises to the top of the lake, and cold, more dense water is at the bottom of the lake. And stratification occurs even in very shallow lakes and ponds. However, it may only last for hours or days. And large lakes like Cayuga Lake here are an example. At this site, it's about 70 meters deep, so over 200 feet. The lake stratifies strongly from June into October. And most lakes in the state with depths greater than 10 meters will stratify fairly strongly. Lakes that have long historic records that we've looked at have been warming demonstrably in recent decades. All evidence suggests that warming will continue for additional decades, leading to stronger stratification, a longer duration of stratification. Stratification starts earlier and ends later in the year. And that will and likely is influencing the structure of various biological communities, including cyanobacteria or green algae. One thing you might notice from this graph are all these stripes or vertical discontinuities. And what that is, that's evidence of satiation, where the lake rocks back and forth. You can see on the map on the left that red dot marks where this profile was taken. So it was taken near the southern end of Cayuga Lake, near the city of Ithaca. And in the next slide, I have a little movie. It lasts for about a minute, and it covers approximately the month of September of 2013. You can see the warmer Ephemeral is the red color on top, and that's rocking above the colder Hypolimnetic in blue. And these internal waves or seiches are set up by wind pushing the warmer water to one end of the lake, and then it then rebounds as a standing wave at the thermocline back to the other end of the lake. And this is a really neat example. We often think of lakes as quiescent, not very turbulent, but here you can see that rocking effect can actually splash cold metalimnetic or hypolimnetic water up onto the shallow shelves at the northern and southern end of Cayuga Lake. This happens to, in all lakes, just in smaller lakes at a much smaller scale. One of the consequences of thermal stratification is depletion of oxygen in the hypolimnetic or in the lower waters. These are plots from Onondaga Lake, experienced a remarkable recovery over decades due to decreases in nutrient loading. And you can see that the amount of red, or the amount of anoxic water, has decreased over time. And that was a consequence of lower phosphorus loading and lower trophic state of the lake. And this resulted in improved conditions, particularly for warm water fish. However, if you want cool or cold water fish, you need a combination of conditions that includes water that's less than about 20 degrees Celsius. So the water temperature needs to be below this blue line, and you need lots of oxygen. Your dissolved oxygen concentration has to be better than 5 milligrams per liter. So you have to be above the red line. And you can see during summer in Onondaga Lake, those two conditions do not coexist. So there is essentially no cool or cold water habitat in Onondaga Lake. And neighboring Atisco Lake, which is similar in many ways, just less productive, lower nutrient loads, you can see that oxygen depletion is not as bad, and there is at least some marginal habitat for cool water fish, including walleye and brown trout during the summer months. We've talked a little bit about lake productivity or trophic state, and this is really all about algal growth. Algae need nutrients to grow, macronutrients such as carbon, nitrogen, phosphorus, they need in large supply. There are also micronutrients that they only need at trace levels. And in limnology, we've all learned about Liebig's law of the minimum. And basically what that says is that the nutrient that's available in the shortest supply relative to the needs of algae will limit their growth. So in marine systems, that happens to be nitrogen. You want to decrease productivity in a marine system, you reduce nitrogen. And phosphorus, the nutrient limiting algal growth, by and large, is phosphorus. That was demonstrated nicely by Dave Schindler and colleagues in the experimental lake area in Canada back in the early 70s. They added carbon and nitrogen to both basins of this lake, only phosphorus to the basin on top, and you can see the result. They produced a massive bloom. Prior to this, there was an argument that carbon limited primary production in lakes. Since this demonstration, our lake management efforts, freshwater systems have largely focused on phosphorus, and rightfully so. And with that, I will pass the control over to Monica.
Thanks, Dave. So here in New York, we are very lucky to have such a great community science program called CSLAP. It is led by the New York State Federation of Lake Associations and the Department of Environmental Conservation. In this long-standing program, volunteers from across the state collect water samples from their local lake every few weeks during the summer, and then send these samples to our lab here at UFI. We analyze the samples, and then the DEC gives the Lake Associations annual reports of their water quality results, and help guide future monitoring and future management. In 2019, we had over 160 lakes participate, and there's even a few more that joined this year. So we just wanted to say thank you to all of our CSLAP volunteers for taking the time to collect and send samples and just being great water stewards in New York. Because of you, we are lucky to have such a unique data set that while not has data for every single lake here in New York, it represents quite many lakes here in the state and allows us to look at our lakes from a bigger picture. Here are a few graphs showing averages of certain lake and watershed characteristics of our CSLAP lakes. On your left is a map with all the CSLAP lakes that participated between 2012 and 2017. On your right are bar charts that show some of the different morphology, morphometry differences between the regions of the state. So in the western region, shown here in the darker green, there are larger, deeper lakes than the rest of the state, which makes sense because this hosts the Finger Lakes, which are some of the deepest and largest lakes here in the state. The southern region has more small, shallower lakes that you can see in the bottom plot tend to be a little bit warmer than the other regions. The western, central, and northern regions typically have similar temperatures. These plots contain information regarding percent land use within the individual watersheds of the CSLAP lakes. The northern and central regions are mostly forested, and this makes sense because the northern region and part of the central region are within the Adirondack Park. The western region typically has more agricultural lands, and as we go further downstate towards New York City, the southern region has on average more than 10% of developed lands within their watersheds. These changes to the landscape, like urban development and agricultural practices, can eventually influence water quality. Finally, we have the three typical indicators of trophic status. Total phosphorus concentrations, shown here in the top plot, were typically higher in the southern and western regions of the state. And if you remember to the previous slide, these regions also had the highest percentages of developed and agricultural lands. Chlorophyll A, which can be used as an alternative way to estimate phytoplankton biomass, was also higher in these regions. Secchi disk depth, or water clarity, was highest in the northern regions, where lakes have a bit less human influence on them. The lakes are home to many aquatic organisms that we may love. The aquatic ecosystem can be broken down into groups called trophic levels, which basically shows us the direction of energy or nutrient flow. Nutrients within the lake and sunlight support our primary producers like phytoplankton and aquatic plants. Then we have consumers. Consumers can't make their own energy and have to eat something else. So these three higher levels basically indicate what eats more meat, like the tertiary consumers, like largemouth bass, versus what eats more plants, the primary consumers. Sometimes animals don't fit necessarily well into these categories because their diets may change during their life history as they age, or they're competing with other animals with similar diets, or there's other environmental stressors affecting them. When members of each of these groups dies, the nutrients are recycled by decomposers, and then any kind of excretions also feed back into the growth of primary production. Some nutrients do get lost by sedimentation or the process of the nutrients settling to the lake bottom, or then flow on downstream to connected water bodies. These interactions between organisms can be quite complicated, and they do respond to water quality and human interferences. Starting at the bottom of the aquatic food web, we have algae. They're very diverse and can live in a variety of habitats, like some are attached to rocks and plants, some are free floating, and some can even look like plants. The free floating algae are called phytoplankton, and they can be broken up into further groups that make up the community in the lakes. In New York, diatoms tend to dominate the lakes during the spring. Then once temperatures increase, green algae tend to become more dominant. Not only do these good algae provide food to aquatic animals, but they also produce oxygen to the atmosphere and to the water column. These groups can form algal blooms that, while not hazardous to human health, can affect the smell and taste of the water, as well as the look and the overall water quality. Then that brings us to the most infamous plankton, blue-green algae. Blue-green algae really aren't even algae at all, but photosynthetic bacteria called cyanobacteria. Harmful algal blooms, also called HABs, are typically dominated by these cyanobacteria species and may contain toxins that are hazardous to our health. Here are a few examples of HABs in New York State from last summer. We may call them blue-green, but as you can see, they don't always have that namesake color. Blooms can contain several species of cyanobacteria as well as some of the good algae. It's difficult to tell the difference between what is dominated by good algae and what's bad. So if you see a bloom, you should just report it to the DEC. You can visit the DEC's HABs website, shown here at the bottom of the screen, for more information about what to do if you see a potential bloom in your lake. Lakes worldwide are actually experiencing HABs more frequently, and New York is following suit. So why is New York turning blue-green? Like green algae, cyanobacteria flourish in nutrient-rich environments, those lakes that have elevated amounts of phosphorus and nitrogen. However, unlike green algae, cyanobacteria can actually withstand hotter temperatures. This may mean that we will be seeing more cyanobacteria and blooms in the coming years as water and air temperatures steadily increase due to climate change. Cyanobacteria also congregate better under calm conditions, and waves can disperse these large accumulations, and then wind can even push them from the surface of open water areas of the lake to the shoreline where we can see them. Another factor that has been more recently discussed is the presence of dracinid mussels, or those invasive zebra and quagga mussels. These mussels may in some cases selectively not eat cyanobacteria and preferentially eat the green algae. And in some cases, if these mussels weren't present, the phytoplankton, rather than being filtered through, may have undergone sedimentation. But as a result of a large mussel population, the nutrients that would have undergone sedimentation are actually recycled into forms that can be used once again by cyanobacteria. Our CSLAP lakes provide an excellent data set that allows us to take a look at HABs here in New York. On your left are two graphs with bars indicating the number of lakes that experienced a bloom during the summer from May to October of 2012 to 2017. The top plot represents open water samples, and the bottom plot represents samples that were collected from the shoreline. Overall, there are more blooms along the shoreline than there are in the open water. With both plots, you can see the number of lakes that experience blooms increase steadily during later summer months, like through July to September, when it's hot and usually a little bit less windy. The shaded part of each bar indicates blooms that were low in toxins, whereas the parts of the bar that have the diagonal lines indicate the number of lakes with blooms that had high toxins. As you can see, there were more lakes with toxic blooms along the shorelines, and the open water samples, there were very few open water blooms that had high toxins. An analysis of influential factors showed that the lakes that had toxic blooms were also likely to have high nutrient levels, agricultural lands within the watershed, and dracinid mussels present. There's still a lot to learn about HABs and how these distributions may change due to climate change and changing landscape. Still within the primary producers category, we have our aquatic plants, or macrophytes. Like algae, they're photosynthetic. They can look widely different and inhabit many areas in and around lakes. Along the shorelines, we have the riparian zone, which often serves as a line of defense and provides some erosion control. Next are emergent plants, and then we go out into the littoral zone for more fully aquatic plants. The littoral zone is the area of the lake where light can get through the water to support plant growth. In some cases, such as very shallow lakes, the littoral zone can make the majority or all of the lake. Submerged plants have most of their leaves underwater. There are some species that can grow all the way up to the surface and then sprout out on top of the surface to shade out their competition. There are also floating plants that only have leaves on the surface and none beneath the waterline. There are floating plants that have rooted, the classic roots we think of going down into the sediments, but there are also floating plants that don't have these classic roots going all the way down to the sediments. All plants are important to the ecosystem as they produce oxygen, can slow erosion, and provide habitat for other animals. But of course, when conditions are just too perfect for plant growth, it can be a lake user's nightmare. Not only do they get caught in boat propellers, they can look unsightly and feel gross against your skin when you're swimming. When a large amount of plants die off, the plants are broken down and they may deplete oxygen levels in the water and eventually could contribute to internal loading of nutrients. Moving up one trophic level are zooplankton and invertebrates, or small animals without backbones. There are three major types of microscopic zooplankton. We have rotifers here in the middle, copepods, and then cladocerans here on your left. Cladocerans tend to be the most efficient grazers of phytoplankton. An abundance of these herbivorous zooplankton, like the Daphnia species, can help actually control algal growth. Zooplankton are not the only invertebrates in the aquatic ecosystem. There's insects, insect larvae, snails, clams, and mussels that can all exist in the shallow and deep areas of the lake. This whole group plays a very important part in regulating nutrient cycling, and they can indicate when there's something else affecting the ecosystem. So I have a local example of these interactions in Onondaga Lake. So during the 60s through most of the 80s, salinity was high in Onondaga Lake due to industrial inputs. During this time, an invasive Daphnia species was more dominant than the native Daphnia species due to the invasive species' tolerance for salty environments. After the closure of the soda ash producing facility in the mid-80s, salinity decreased, as you can see here in this top plot. And the native Daphnia were able to start recolonizing the lake. Seki depth during this time was pretty variable during the 70s, but as you can see, there were some years that had higher Seki depth than others. And these years were marked with clear water phases. Clear water phases are periods of high clarity, typically due to these Daphnia species grazing on phytoplankton. During times when Daphnia are high, like here over on this graph, the biomass gets high, they can effectively mow down the plankton, which is signified here by our chlorophyll A measurements. You can see as the biomass goes up, the chlorophyll goes down, and they were able to create this clear water, which was indicative by this drop in Seki depth, well, increase in Seki depth rather. You can see further down in the lake. So during the 80s, Seki depth was pretty low as these native Daphnia were bouncing back. And then through the mid-80s to the 2000s, there were a series of upgrades made to the local wastewater treatment plant that flows into Onondaga Lake. And this effectively reduced phosphorus levels, shown here in the middle, from being hypertrophic in the orange down to mesotrophic levels, shown here in the yellow. During this time, you can see Seki depth greatly improved and clear water phases were frequent and occurred every single year. Then, all of a sudden, clear water phases stopped in the mid-2000s. And you would think less phosphorus means less phytoplankton, right? That's what you've always heard. Well, there was another player in this game. Invasive algal became dominant during this time due to these large improvements and were efficient predators of the Daphnia. So the Daphnia population couldn't get to these large numbers and create these clearing events. In 2008 and 2009, clear water phases occurred again in response to a massive die-off in the alewife population. However, the clear water phases have not occurred again since 2010, which may be due to the alewife population rebounding or other dynamics within the biological communities. With that, I'll hand it off to Dave A.
All right, thank you, Monica. So fish are an integral part of any lake, and we all are familiar with their recreational importance. However, they do play an important role in the ecology of a lake as well. Monica just illustrated the importance of that invasive alewife. They typically influence a lake via their trophic importance, or what they're eating or what's eating them. Additionally, they can act as harbingers of change. Dave mentioned earlier the example of cold water fish species in Onondaga Lake being forced out of their thermal range and not existing in Onondaga Lake. So the disappearance of certain species can indicate a change in water quality. Other large animals utilize lakes as well. We're all familiar with the impact that a large population of geese can have on a body of water, both in terms of the aesthetics as well as in terms of water quality. But it's important to note that it's not all bad when we talk about waterfowl. They do graze on macrophytes, which can be considered nuisances in some cases. And of course, it's just enjoyable to have some waterfowl floating around your lake to enjoy. Beavers are well-known ecosystem engineers that impact lakes by raising water level rather, either in the lakes directly, or they can dam up tributaries and create impoundments in the watershed, as is seen in the top photo here. They effectively altered the landscape by creating these impoundments, and you can see they will kill these, on occasion do kill trees and things like that. And these dams can be breached occasionally by natural processes or through human intervention if they're causing upstream problems. And this can introduce a large amount of water and sediment and nutrients all at once into the lake, which can be important depending on the timing of that input. And there are a bunch of other organisms that utilize lake environments, and I just include salamanders here. I think they're a really neat example of an animal that has both aquatic and terrestrial life stages, and they're a decent indicator of water quality because they utilize the benthic habitat, which can become degraded faster under degraded water quality conditions. So now I want to jump out of the lake and into the watershed. I've worked quite a bit in the Owasco Lake watershed over the past year for a project that UFI is working on. So I'm going to use that as an example. So we have a map here on the right of the entire Owasco Lake watershed with land cover use overlaid on it. Those are the myriad of colors that you see on that map. In addition, you can make out some black lines within the watershed. Those are actually tributary sub-watersheds, so each individual stream that is draining directly into the lake. The scale of a watershed is very important when we're talking about lakes. So Dave touched on this earlier with the example of Black Lake compared to Skinny Atlas Lake. And Skinny Atlas Lake is a really great example of a small watershed to lake area ratio at about 5:1. Whereas Owasco Lake has a much larger watershed relative to the lake area at about 20:1. So the impacts from the watershed, or the inputs from the watershed, are going to be much higher, much more important to the water quality of Owasco Lake than in Skinny Atlas Lake. And even though it is, in fact, Skinny Atlas Lake's neighbor. Another consideration when we talk about scale is the size of the receiving water body. So the photo on the left is of a power plant on the shores of Cayuga Lake. This power plant utilizes lake water for cooling, and UFI did a study a while back that found essentially no net impact on the overall thermal budget of Cayuga Lake from this power plant. However, you could imagine if this same power plant was on a much smaller lake, the impacts could be quite profound. Now, if you look back and forth between these two images, these two maps, you can start to get an idea of how we start to determine watershed boundaries. And this process we call delineation of watersheds. So on the left here, you can see elevation contours. And where they get really tight together, They form really dark areas of the map here. Those are very steep areas of the watershed. So you can start to get an idea of how the streams actually flow, even though I don't have the streams overlaid on this map because I figured there was already enough lines here to make people dizzy. You can certainly tell that water would flow down these low portions of the watershed. Something else you might notice is this large area of flat here at the southern end of the lake. This is associated with this light blue area here on the land cover map. This is an area of wetlands known as the Owasco Flats. Wetlands act to slow down tributary flows. They increase sedimentation, and they can increase nutrient uptake during periods of the year when primary productivity is high. This is a process we call attenuation. Something else you might notice about the land cover use in the Owasco Lake watershed is that it's highly agricultural. These are the yellows and browns in this map. You might also notice that there is a higher percentage of agricultural land in the northern portion of this watershed than the southern portion, which is entirely made up of a single tributary watershed, the Owasco Inlet, which is responsible for over half of the hydrologic inputs or flow inputs to Owasco Lake. So when we're talking about loading, even though it's very important, again, the scale, the Owasco Inlet's putting a lot of water and a lot of compounds into the lake. Some of those smaller watersheds to the north might be more important or easier to manage because they have a higher percentage of agriculture in them. And when we look at loads this way, we look at them on an aerial basis. So in Owasco Lake, you can see here the watersheds line up from largest to smallest in terms of the big four that are named: the inlet, Dutch Hollow Brook, Sucker Brook, and Vanessa Brook. And then the minors make up all these small watersheds that don't have names on this particular map. Some of them are named. So the inlet on an aerial basis actually has the lowest impact in total phosphorus in kilograms per hectare being about 2.5 acres, whereas the two smallest watersheds actually have the highest aerial loading rates here, with Sucker Brook over here and Vanessa. And this starts to make sense when you realize that Vanessa Brook has about 80% agriculture, and the inlet is actually less than 50% agriculture. So the character of compounds entering the lake is important, and when I talk about character, I'm talking about the form in which compounds exist. They can be particulate or dissolved. Particulates, like in the top photo, aren't always associated with an increase in algal biomass unless they're accompanied by dissolved, more bioavailable forms for algae to uptake immediately. I mentioned on the previous slide areas of value such as wetlands and the importance they have for attenuation. Areas of concern might be areas of tributaries that have high stream banks that are highly erodible, or areas with very little or no vegetated buffer along tributaries or directly along the lakeshore. And then the type of lake that's receiving watershed inputs is important as well, especially in terms of its flushing rate, which Dave mentioned earlier, the rate at which the water in a lake actually turns over and becomes new. If you have a very long flushing time, you can effectively shut off the spigot, so to speak, of watershed loading, and you won't see a respective change in lake water quality for a long time if you have a long flushing time, a long flushing rate. Whereas a lake that turns over multiple times per year will respond much quicker to changes in watershed loading. So to just kind of pull all these watershed ideas together, I'm presenting the case study of acid rain. Acid rain was a huge problem over the past couple of decades in New York State, and it originated from industrial pollution in the Midwest, mostly sulfur and nitrogen compounds released into the atmosphere. They were carried over New York State by the prevailing westerly winds, and then the precipitation fell on New York State with a decreased pH, a very significantly decreased pH historically, as low as 4. And in the northern portion of New York, areas like the Adirondacks, the soil and bedrock has a very low acid neutralizing capacity or capability to buffer that acid rain. And the lakes were significantly impacted in those northern regions. Whereas in the central and southern portions of New York, the acid rain still fell, but it was buffered by soil and bedrock containing a lot of limestone, which prevented those deleterious impacts on the lakes. So it's just a nice example of how the geology, weather, human uses all come together and have a different impact depending on where you are in the state. And I'm happy to say that acid rain has become less of a problem because of things like the Clean Air Act. And you can see on this bottom map the decrease in acid deposition over time. So to just wrap up here today, this mind-opening day of limnology, or morning of limnology, I want to talk about management a little bit. And there's plenty of resources out there that you can find that will walk you through any management scenario you can imagine. So instead of boring everybody and putting them to sleep with hypotheticals, I'm going to talk about a couple of case studies, but I just wanted to mention there's multiple options when it comes to management. You can go in lake, you can do things in the watershed. There's short term versus long term. Some things are going to have a quick result, but they might not have the staying power of the long term efforts. And of course, cost is always a concern. It's really important that you understand your lake and you set realistic expectations for your given lake, because each body of water is unique. So the first example I'm going to present is the case of external loading in Onondaga Lake. Onondaga Lake is near and dear to UFI's heart, if you couldn't tell from our multiple examples already, but it also stands as a great example of lake restoration. Onondaga Lake is typically associated with industrial pollution historically, but the role that cultural eutrophication has played in its history is just as important. Here we can see an aerial view of Onondaga Lake and its setting next to the city of Syracuse. And you can see the wastewater treatment plant down here at the southern end of the lake, which actually discharges directly into the lake itself. And as Monica mentioned, over time, the effluent from that wastewater treatment plant has been improved through upgrades to the treatment process and decreased the amount of phosphorus from really extremely high levels above 2 or 3 micrograms or milligrams per liter down to levels that are well below what many of the wastewater treatment plants in New York State currently are required to discharge effluent at of 1 milligram per liter. Metro in Syracuse currently discharges below 100 micrograms per liter. So that decrease has actually led to a rebound in Onondaga Lake and a shift from severe eutrophy to upper mesotrophy. And that is directly concurrent to that load reduction in the wastewater treatment plant. Now, it's interesting to note that these reductions, as shown here in these bar graphs with those 3 indicators of trophic level, total phosphorus, chlorophyll A, and Secchi depth, are very similar to those that we're seeing out in Lake Washington in Seattle, near Seattle. And if you're not familiar with that case study, that wastewater effluent was actually diverted around the lake. So in Onondaga Lake, the wastewater effluent makes up actually like 20% of the annual flow budget for the entire lake. And still, just with improvements in technology and wastewater treatment, we were able to achieve very similar results and recovery of the lake. So the second case study I'm going to present is top-down management in Otsego Lake. So Otsego Lake near Cooperstown, New York, had an introduction of invasive alewife around 1987, which is right about here. And following that, you can see a precipitous decline in water clarity. This was also associated with an increase in oxygen demand, which forced cold water fish like whitefish and lake trout out of their preferred ranges thermal ranges, and they were nearly lost. The whitefish were, and lake trout had a bunch of problems as well. This was due to those alewife, as Monica talked about briefly. They were grazing on those large zooplankton, which allowed algae to proliferate. In 2002, a walleye stocking program was implemented in an attempt to control the alewife population. On the bottom here, you can see the alewife catch in trap nets around the lake. So walleye stocking began here in 2002, and the program worked actually better than expected. And today, alewife are essentially extirpated from Otsego Lake. I don't know when the last time was they actually observed one in the lake. So this is a good example. Typically, we think of bottom-up management when we're talking about water quality management. Decrease these nutrients going in at the bottom, and you decrease the primary producers. But in this case, the problem was that the secondary consumer of the alewife was grazing down those primary consumers, allowing the algae to explode. The addition of the tertiary consumer, the walleye, reduced the secondary consumer population and brought everything back into balance. So just to summarize everything that we've covered today, to understand basic limnology requires that you understand essentially everything that we've talked about and many other topics that we didn't have time to touch on today. But understanding the physics, the geomorphology of your lake, the chemistry, how things act and interact in water, and then biology, which is essentially a result of the physics and chemistry of the lake, is important. Effectively managing aquatic ecosystems requires that you set realistic goals for your lake through an understanding of your unique body of water. It requires collaboration among local and state authorities and effective education and outreach. So with that, I want to say thank you for attending. Thank you for your attention. And I believe it is time. If anybody has any questions, I will turn it back over to Savannah.
Thank you all so much. So all I'm going to do is switch over to one slide here. We have received a ton of questions. So I just want to reiterate to folks on the call, in case we are not able to answer your question live, we will be following up with you via email after this presentation. So for the first question, are there any native plants which can be grown to replace invasive vegetation? And are there any new invasive plants or animals impacting lakes that are on Upstate Freshwater's radar?
So this is Dave Andrews. In terms of the plants that might be able to replace invasives, it certainly is a management option. And I did a little bit of research during my master's work that suggested if you have very dense invasive plants, if you can get them kind of beat back and give the natives a chance to repopulate, you can expect to see about a 50-50 regrowth between the invasives and the natives. Typically, you won't be able to completely take over an invasive species bed with a native, but there is some room there, and there's some evidence to show that if you are able to use maybe a benthic mat or dredging or hand pulling, that the natural seed bank that exists can be given a chance to rebound a little bit.
Great. So moving on, how, and I do apologize if I mispronounce this word, how does seiching wave motion at the surface compare to natural convection of warm to cold water at the bottom?
Yeah, I think that really gets down to the difference between advective motion and diffusion. And really, physical advective motion can move much more water, transport much more maybe nutrient-rich, colder water to the surface more rapidly than could natural diffusion, which is a very slow process. So yeah, seiching is really vital to the mixing of lake waters.
Great. And we had another question that asked if you can talk about why conductivity is an important metric and what it means.
Well, yeah, conductivity is an indicator of salinity or the dissolved solids in water. And freshwater organisms have really evolved to live in a relatively narrow range of conductivity or salinity. And when salinity gets elevated, as Monica discussed in Onondaga Lake, that can structure biological communities, for example, zooplankton or fish. They may not be able to survive in a saltier environment, therefore structuring communities and maybe impacting water quality, algal growth, for example. Another issue with salinity or conductivity is it can enhance thermal stratification and make water harder to mix potentially.
Perfect. And then, so moving into algal blooms, we've received a ton of questions about that. So does removing all of the macrophytes in a lake lead to harmful algal blooms?
Well, that's a tough one. There is some competition for nutrients between macrophytes and phytoplankton. There is the potential for that. There is some evidence that lakes where they have removed macrophytes have seen increased blooms. I don't think we can say that that is necessarily always the outcome. I think really what we search for is some balance, right? Where you need macrophytes, they're critical. They can help to compete with phytoplankton to some extent, but we just can't live with the just explosive overproliferation of macrophytes.
Thank you. And so another attendee asked, they have been seeing large amounts of suspended algal that are not in mats or rafts. Is that a precursor to a sort of bloom? And does a bloom need to be concentrated in a mat or raft form to be harmful?
Yeah, well, yeah, definitely not. Definitely blooms can occur. They can look just, maybe like streaks in the water. Sometimes the water might just appear grainy. There are large colonies of cyanobacteria that can aggregate. So, yeah, kind of the scums that form along the shoreline are an extreme example. It's really tough to speculate in terms of the suspended phytoplankton, what that might be. I'd recommend perhaps collecting a sample. You could send it to a place, we could look at it for you and let you know what it is. That might be the first step.
Great. And then one more on algal blooms. Can algal blooms move up and down in the water column based on their own control or devices?
Yes, certain species can. For example, Microcystis, which is the most dominant cyanobacteria in New York State, does have quite an ability under calm conditions to migrate up and down in the water column over meters. So what they can do is during the day when the sun is out, they can hang out up in the upper layer and take advantage of sunlight and warm water. And then at night, they can migrate down to more nutrient-rich water, or even down near the bottom of a lake and load up on the nutrients. Yes, that definitely occurs and can be important.
Thank you. So we do have, a few more moments for a few last questions, but I did just want to highlight that in the chat box, we have provided a Zoom link. And if you would like to join us for half an hour of networking at the end of this webinar, please click on that link and join us at, 11 o'clock. You can call in using the phone number displayed, or you can use the link that was sent through the chat box. So we'll have time for just two more questions here. Can attendees, is there a way for them to receive a photo or a diagram of their specific lake watershed?
Yeah, so the best way, there are actually some online tools, and I'd be happy to communicate via email if whoever asks this question wants to reach out. But you can get some kind of quick and dirty maps of your watershed and certain things like the maps that I showed today. But to get into the really more technical maps, we use GIS software, and you have to have some experience with that. Yeah, I've developed a little bit of that over time. There's certainly folks that are way better at it than I am. So it all depends on what you're looking for, but you certainly can get an overall picture of what your watershed looks like by through a couple of online tools that I'd be happy to talk about with anybody who's interested if they want to reach out.
Great. We will definitely be following up after this webinar because there are quite a few questions that we were unable to get to live right now. So I do just want to thank all of you for being here today to provide your expertise for this presentation. And I would just like to highlight again that the UFI experts will be joining us on the Zoom call of the networking session that is happening. You know, we're going to jump right over to that. So please join us. That link is provided in that chat box. You know, send it back over to the UFI folks for any remaining closing comments.
Thanks. It was great. Hopefully we can do it in person next time.
Definitely. So for folks on the call, I'm going to be leaving this webinar session open just so you can grab that link in the chat box if you're interested for joining us. But, you know, thank you to Monica and both Davids here with us today again for providing this expertise. And we'll be following up with folks with those unanswered questions.